Welcome to another episode from the Veterinary Research Channel. Today, we delve into a fascinating study that sheds light on the complexities of severe asthma in horses, a condition that significantly impacts their well-being and athletic capabilities. This research, conducted by Laurence Leduc and colleagues at the Université de Montréal in Canada, investigates the role of nerves in the airways of horses suffering from this chronic respiratory disease. Severe asthma affects a notable percentage of adult horses, around 15%, leading to a diminished quality of life, reduced athletic performance, and, unfortunately, in some cases, premature retirement or euthanasia. The primary cause of breathing difficulties during asthma flare-ups in horses is the narrowing of the airways, known as bronchoconstriction, and an exaggerated response of these airways to inhaled environmental triggers, a phenomenon called airway hyperresponsiveness. Interestingly, administering bronchodilators, medications that help relax the airway muscles, often leads to a substantial improvement in lung function, sometimes as much as 60 to 70%. This highlights the significant role of airway muscle contraction in the disease process. Beyond these acute episodes, there's also evidence of a persistent, albeit milder, airflow limitation in horses with asthma, even when they are in periods of clinical remission and exposed to fewer environmental triggers. Even in these calmer phases, bronchodilators can still have a positive effect, suggesting an ongoing underlying issue with airway tone. Furthermore, a common symptom in asthmatic horses, coughing, is itself a neural reflex, indicating the involvement of the nervous system in the disease. Intriguingly, airway hyperresponsiveness can sometimes be observed even when there's no active inflammation in the lungs. This observation suggests that other mechanisms, apart from inflammation alone, might be contributing to the development and persistence of this exaggerated airway reactivity, and nerve activity is one such potential factor. Research in humans and mice has shown that the sensitivity to inhaled allergens and the contraction of the smooth muscles surrounding the airways are largely controlled by sensory nerves, including myelinated A fibers and unmyelinated C fibers, as well as efferent, both myelinated and unmyelinated, parasympathetic nerves in the airways. It's hypothesized that disruptions in this intricate airway nerve network could contribute to airway hyperresponsiveness, bronchoconstriction, and even the structural changes in the airways known as airway remodeling. For instance, studies in mice have indicated that acetylcholine, a neurotransmitter released by nerve endings, can increase the mass of airway smooth muscle. Moreover, in dogs, a surgical procedure called vagotomy, which involves cutting the vagus nerve, has been shown to prevent airway hyperresponsiveness and inflammation. This further underscores the importance of neural pathways in respiratory health and disease. The impact of altered nerve function might not be limited to the bronchi themselves. In humans with asthma, an increased presence and branching of nerves containing substance P, a signaling molecule, have been observed not only in the airways but also in the blood vessels and glands of the lung tissue. The nerves of the respiratory system, both those that send signals to the brain and those that carry signals from the brain, primarily travel via the vagus nerve. These nerves are crucial in regulating not only the tone of the airway smooth muscles and breathing patterns, but can also influence blood vessel tone, mucus production, and inflammation through receptors called muscarinic receptors found on various cell types. Furthermore, it's believed that this network of nerves might be involved in the remodeling of both the airways and the blood vessels that occurs in horses with severe asthma. While there is evidence suggesting functional changes in airway innervation in horses with severe asthma, a detailed histological examination, meaning a microscopic study of the nerve structures in the airway walls, had not been thoroughly investigated until this research. Therefore, the aim of the study was to carefully characterize the nerves surrounding the airways in horses with asthma using specialized techniques called immunohistochemistry and histomorphometry. The researchers hypothesized that both the number of nerves and the total area occupied by these nerves would be greater in horses with severe asthma compared to healthy control horses. Given that the primary target of these peribronchial nerves is the smooth muscle that encircles the bronchi and smaller bronchioles, they also specifically looked at whether the innervation associated with this smooth muscle was increased. Additionally, to determine if any observed increase in nerve presence was specific to the airways, they also examined the nerve supply to the walls of adjacent pulmonary arteries. To conduct their study, the researchers utilized lung tissue samples collected after death from 8 horses diagnosed with severe asthma and 8 healthy horses. These samples were obtained from the Equine respiratory tissue biobank. For each horse, detailed information regarding their lung function before death was available, as well as the results of a bronchoalveolar lavage fluid analysis, which provides insights into the cells present in the airways. The history of the horses' housing conditions and diet was also reviewed. The horses with severe asthma included some that were experiencing an active flare-up of their asthma at the time of euthanasia, and others that were in a period of remission. The healthy control horses had no history of asthma or respiratory problems and exhibited normal lung function and airway cell profiles. The researchers employed a technique called immunohistochemistry, using a specific antibody that targets a protein called S100, which is found in Schwann cells, a type of cell associated with both myelinated and non-myelinated nerve fibers. This antibody essentially acts as a marker to visualize the nerves in the lung tissue under a microscope. The number and the cumulative area of these identified nerves in the region surrounding the airways, as well as those closely associated with the airway smooth muscle, were then carefully measured using a process called histomorphometry. These measurements were adjusted to account for differences in airway size, allowing for a more accurate comparison between different samples. The results of this detailed analysis revealed some significant differences between the horses with severe asthma and the healthy controls. The researchers found that both the number of nerves and the total area occupied by nerves in the tissue surrounding the bronchi were significantly higher in the horses with asthma compared to the control group. Specifically, the median number of nerves in the peribronchial region was considerably greater in the asthmatic horses, and the cumulative nerve area was also substantially larger. Furthermore, when the researchers specifically looked at the nerves located within or immediately adjacent to the airway smooth muscle, they again found a significant increase in the number of these smooth muscle-associated nerves in the horses with asthma. While the total area of these smooth muscle-associated nerves also tended to be higher in the asthmatic group, this difference did not reach statistical significance. In line with previous research findings, the study also confirmed that the area of the airway smooth muscle itself was larger in horses with severe asthma compared to their healthy counterparts. This muscle thickening is a characteristic feature of airway remodeling in asthma. Interestingly, when the researchers examined the pulmonary arteries, they did not find a significant difference in the number of nerves supplying the walls of these blood vessels between the asthmatic and control horses. However, similar to the airway smooth muscle, they did observe that the vascular smooth muscle area in the pulmonary arteries was increased in the horses with severe asthma. Finally, the study explored whether there were any correlations between the increased nerve presence and lung function parameters or the degree of airway inflammation. However, they did not find any significant correlations between the number or area of airway nerves and the measures of lung resistance or elastance in the horses with asthma experiencing an exacerbation. Age was also not found to be a significant factor influencing nerve density within either the asthma or the control group. The findings of this study provide novel insights into the role of airway innervation in equine asthma. The observation of an increased nerve supply to the peripheral airways in horses with severe asthma suggests that this might be an important factor contributing to the severity of the disease. This increase could manifest as either a greater number of individual nerve fibers or an increase in both the number and size of these fibers. However, the specific techniques used in this study did not allow the researchers to definitively distinguish between these possibilities. The intricate network of nerves in the airways and lung tissue plays a crucial role in regulating the tone of the airway smooth muscle, influencing inflammation, and controlling mucus secretion. Therefore, any alterations in the structure or function of this innervation could lead to disruptions in these vital processes. For example, sensory nerves in the airways are known to play a significant role in triggering airway smooth muscle contraction in response to various stimuli. An increased nerve supply could potentially lead to an exaggerated contractile response, contributing to airway hyperesponsiveness. Furthermore, the effectiveness of anticholinergic drugs, which block the action of acetylcholine, in dilating the airways of asthmatic horses highlights the role of cholinergic nerves in bronchoconstriction and mucus production. The persistence of airway hyperresponsiveness even during periods when inflammation is minimal further supports the idea that neural mechanisms are involved in maintaining this heightened airway reactivity. Indeed, studies in other animal models, such as dogs and mice, have demonstrated that blocking or cutting specific nerves can reduce or even abolish airway hyperresponsiveness. Prior research in horses has also indicated that blocking vagal nerve activity can reverse some of the physiological changes associated with airway obstruction. The current study also found an increase in the number of nerves associated with the thickened airway smooth muscle in asthmatic horses. This observation supports the hypothesis that there is a link between nerve supply and the structural remodeling of the airways, specifically the increase in smooth muscle mass. However, the study's findings do not definitively establish whether the increased innervation is a cause or a consequence of this smooth muscle remodeling, or perhaps a combination of both. It's known that during development, the maturation of airway nerves closely follows the development of smooth muscle. Therefore, an increased nerve supply might be a compensatory mechanism to manage the increased number of muscle cells in asthma. On the other hand, evidence from other systems suggests that nerve signals can influence the differentiation and growth of smooth muscle cells. While sympathetic nerves have a limited direct role in controlling airway diameter in most species, cholinergic nerves have been implicated in smooth muscle remodeling. For instance, studies in mice have shown that anticholinergic drugs can reduce airway smooth muscle mass. The observation of one control horse with unexpectedly high airway innervation and smooth muscle, despite meeting all criteria for a healthy horse, raises interesting questions. It could suggest a baseline relationship between smooth muscle amount and innervation, regardless of asthma status, or it could even indicate an undetected mild form of asthma in this particular animal. Beyond their direct effects on airway smooth muscle, peribronchial nerves can also influence pulmonary inflammation. Inflammatory cells possess receptors for acetylcholine and can be affected by this neurotransmitter released from airway nerves. Conversely, inflammatory processes can also impact the nerves themselves, leading to their remodeling. It's been shown that inflammatory mediators present in the airways of horses can increase the release of acetylcholine at the neuromuscular junction and enhance the sensitivity of smooth muscle to this neurotransmitter. Furthermore, inflammatory molecules like histamine and leukotriene can increase the underlying cholinergic tone in both central and peripheral airways of horses. This complex interplay suggests that airway innervation can contribute to inflammation and be simultaneously altered by it, both functionally and structurally. The study also confirmed a previous finding of increased vascular smooth muscle area in the pulmonary arteries of horses with severe asthma. However, the lack of a corresponding increase in the innervation of these pulmonary arteries suggests that the increased nerve supply observed in the airways is specific to the bronchial walls and that the remodeling of the vascular smooth muscle is likely driven by other factors, such as chronic low oxygen levels or inflammation. These conditions can promote the migration and proliferation of vascular smooth muscle cells. The researchers also acknowledge that the relatively small sample size might have limited their ability to detect a statistically significant increase in pulmonary artery innervation. The researchers utilized an antibody targeting the S100 protein, a general marker for Schwann cells associated with peripheral nerves. While this is a useful marker for identifying nerves, it does not allow for differentiation between autonomic and sensory nerve fibers. Future research using more specific markers for neuropeptides released by sensory nerves, such as substance P or calcitonin gene-related peptide, could provide a more detailed understanding of the functional neural activity in the airways of asthmatic horses. Although another commonly used pan-neural marker, protein gene product 9.5, was considered, it unfortunately yielded poor staining results in this study. The combination of morphometric tracing and point-counting analysis proved valuable for quantifying both the number of nerves and their cumulative area, especially given the tortuous and small nature of the nerve fibers. The researchers acknowledged several limitations to their study, including the relatively small sample size, which was dictated by tissue availability. Additionally, the asthmatic horses included both those in active exacerbation and those in remission, although the researchers considered it appropriate to group these samples given the chronic nature of the disease and the likelihood of persistent structural changes in innervation. Furthermore, all tissue samples were collected from a specific region of the lung, the caudo-dorsal field, and might not fully represent changes occurring in other lung regions. It's also important to note that horses with milder forms of asthma were not included in this study, limiting the generalizability of the findings to that population. The quantification of nerve structures can be challenging and susceptible to sampling errors inherent in manual analysis. Finally, the point-counting method used to estimate nerve area, while useful for comparing groups, should not be interpreted as an absolute measure of individual nerve size. In conclusion, this research provides compelling evidence for an increased innervation of the peripheral airways in horses with severe asthma. This finding suggests that dysfunction of the bronchial nerves could play a significant role in both the initiation and the persistence of this chronic respiratory disease in horses, potentially contributing to the increased severity of asthma symptoms. To the author's knowledge, this was the first study to specifically investigate the histological characteristics of airway innervation in equine asthma. Thank you for listening to the Veterinary Research Channel. A link for the referenced paper is in the video description. Remember to test your knowledge by answering the questions in the description, and as always, keep those minds inspired, hearts light, and tails wagging.